Pharmacogenetics and pharmacogenomics refers to the study of genetic variants that affects drug response. The patients are always the main focus. The mission for most scientists is to help the patient overcome his or her disease and thus hopefully improve their quality of life. With pharmacogenetics and pharmacogenomics, there is a possibility of improving drug choices by creating safer and more effective dosing options. This also helps researchers and scientists to make improvement in drug development, which in turn help to decrease healthcare costs and also provide better vaccines. Research has shown that genotype variation between patients is a major barrier to treatment. People with the same diagnosis all have varied response to the same drug. When ingesting a drug, bodies of some patients do not have the ability to metabolize the drug in the span required to produce the desired effect. When this is so, and dosing frequency is not altered, there will be a buildup of the drug in the body. Hence, this will cause side effects. If not spotted or treated, it may lead to severe consequences. On the other hand, when ingesting the same drug, bodies of some other patients have too much of this metabolism ability and break down the drug too quickly, eliminating the drug before it even gets the chance to work at an effective level. So why are there two scenarios? This is because there is a presence of the wrong gene. Hence, genetic testing was designed to predict how well a patient's body response would be towards a drug, based on their genetic makeup. The doctor can use your genetic profile to predict if a medication can benefit you or harm you before you even take it. This ensures that the appropriate drug is given, so that it is safe and effective in the shortest time possible. So what are the different types of genetic testing? There are two approaches, the candidate gene approach and genome-wide association studies. The candidate gene approach targets patients with the same diagnosis. They examine the differences of targeted genes by sequencing. Genome-wide association studies also target patients with the same diagnosis. However, they compare the frequencies of single nucleotide polymorphism arrays, also known as SMP, through the genome, as it is also a possible factor which can affect drug response. SMP refers to the nucleotide at a specific position in the genomes that varies in different individuals. For example, a patient with the first combination of genes will have an excellent drug response. For the patients with the second combination, he will have a good response, and for the patients with the third combinations, there will be no effect, while the patients with the last combinations will have a toxic drug response. The major aim of pharmacogenetics and pharmacogenomics is to provide the right patient with the right drug at the right dose. The drug should have the greatest efficacy and least toxicity. Different medication prescribed for different patients is known as individualized therapy. There are also different kinds of cancer drugs. An example is the cancer chemotherapeutic alkylating prodrug, cyclophosphamide. This drug requires cytochrome 450, which is also known as CYP450. It is an enzyme that helps to metabolize the drug before it can work on the cancer cells. Certain patients have malfunctional CYP450 due to the mutation in the DNA. They do not have the ability to process cyclophosphamide. These drugs will in turn be unable to treat these cancer patients with this kind of mutation. In conclusion, all doctors' vision is to provide effective treatment and maximize patients' recovery rate. They hope that in the future, patients with the same diagnosis can recover from their disease with a specific drug that's manufactured for their specific genotype.